Hello, welcome back. This is Professor Young, and this is our beginning look at the nervous system. We're going to spend probably about the next hour or so just kind of looking at and getting acquainted with the overall structure of the star, if you would, of the nervous system. That is the neuron, and kind of applying what we've already learned during our look at muscle physiology, and that is action potentials. Same old story, new location. And so there's not really a whole lot of new learning that's going on. It's just applying what we've already learned to a different situation. And that is something that we will see time and time again in anatomy and physiology. And so with that, why don't we go ahead and jump right on into this and start our look. Really, these first two slides here that we looked at in lecture go back and deal with the overall basic anatomy, if you would, of a neuron. And what you're looking at right here is what we refer to as a multipolar neuron. It is your everyday, run-of-the-mill, typical type of neuron. These are the type of neurons that would connect and innervate with, let's say, skeletal muscle and cause an action potential or a muscle contraction. And so this is ideally when we talk about a neuron, this is the image that pops into most people's heads. And we can see right here, we've got some key structures. The first key structure that we need to consider is the cell body. This is the processing center of the neuron. It is where messages will be received. It's where the messages will be determined as far as what the action is. And then it'll determine when the response needs to be sent. And so it is the brain, if you would, of the neuron. Surrounding that neuron are a series of little branch-like structures, such as these guys right here. And these branched-like structures right there are what we call dendrites. And dendrites are designed to receive a stimulus. They receive an impulse, and they send that impulse towards the cell body. They send that information or that stimulus or that impulse to the cell body. Once that impulse has been received by the dendrites and sent to the cell body, before the message is sent for a response, there's this little area right beneath the cell body, right above the structure that we refer to as the axon, right there. Between the cell body and the axon is the structure referred to as the axon hillock. And the axon hillock is simply the area that joins the cell body with the axon. It is where the action potential is generated, and from the axon hillock, we travel down this long, modified dendrite known as an axon. And the axon is designed to carry the impulse away from the cell body. It carries that impulse to the intended target tissue. And so if it's the case of muscle contraction, the axon would carry the action potential away from the cell body to the skeletal muscle. And the signal, the action potential that it would be carrying would be one that would initiate muscle contraction. Now you will notice that there's some other gobbledygook that is surrounding the axon. The axon here is that long yellow extension. Right there. And there's some other structures here that we need to be aware of. Specifically, that blue structure that is right there, and you'll notice that there are several of them going down the axon. That blue structure that is traveling down or wraps around the axon is what we refer to as the myelin sheath. And if you look even closer, you can tell that in between the myelin, there are gaps. There are gaps between the myelin. Those gaps are what are referred to as the nodes of Ranvier. Now, myelin, it's the axon. And as we're going to find out, this actually helps to propagate the action potential. It helps to travel, get that action potential down the length of an axon very quickly. The node of Ranvier serves as a regeneration station, if you would. And basically it allows sodium ion to rush in. And we'll talk more about this here in a couple of seconds. But those nodes of Ranvier serve a very specific function, and that is action potential regeneration or action potential strengthening. Think of the myelin sheath, if you would, as being the orange casing around the extension cord. And think of the axon as being the wires that's within the extension cord. That myelin is there to keep the electrical current in. Because if it leaks out, you get a shock. Or the object that's being plugged in shorts out. And that's exactly what happens in a neuron. If we start leaking this electrical energy, you short out. And whatever that neuron is going to is not going to be functioning properly. And so the myelin is critical. The nodes of Ranvier are critical. They need to be able to work together. And we'll look at how that happens. And then we've got the end, right down here, the ends of the axon, or what we refer to as the axon terminals. Sometimes you'll see these referred to as terminal buds, synaptic end bulbs. You'll hear that there's quite a few different names that those terminals can go to. But that's the site of innervation. That's what's going to be hovering right above the intended tissue, skeletal muscle, whatever it is. And that's what's going to release the neurotransmitter. To go ahead and either cause a response or pass information on to another set of neurons. There's two choices for those axon terminals. Now that I mentioned it, I'll make it perfectly clear because I don't want anyone confused on that part. Axon terminals can innervate with tissue, right? Skeletal tissue, muscular tissue, other glands, anything like that. Or those axon terminals, those terminal buds can connect to other dendrites. They can connect to other dendrites. And so we've got a couple different options here as far as what can happen. If those terminal end bulbs connect with other dendrites, then what we're doing is we're continuing the sending of the signal. And so The axon terminals will go ahead and make that connection with a dendrite and pass that signal on, which will go down to the cell body, which will then continue to travel down that axon. And so we can link these neurons in series, so to speak, as we travel along. This here just kind of shows you, again, your key structures. You see the cell body, you see the dendrites, the axon, that very long extension coming off the bottom of the cell body. And you've got here they're called terminal buttons, whatever you want to call them. Axon terminals, terminal buttons, terminal end bulbs, synaptic end bulbs. It's all the same gobbledygook. All right. The key thing here is you've got the myelin sheath with the axon that's running through it. Notice the gaps. Those nodes are on Vier. And notice the direction. Everything moves away from the cell body once we get into the axon. All right. So, what is the myelin sheath anyway? The myelin sheath is, again, a insulative structure. It's made of both lipids and proteins, and we have two main types within the body. We have oligodendrocytes, and we have neurons. That's the proper name. The slang term for neurons are the Schwann cells. Oligodendrocytes are a cell that forms the myelin sheath, but only within the central nervous system. Oligodendrocytes are cells that form the myelin sheath specifically within the central nervous system. Now, the central nervous system is defined as the brain and the spinal cord. Schwann cells are cells that form the myelin sheath specifically within the peripheral nervous system. The peripheral nervous system is everything else outside of the brain and the spinal cord. It's everything else. What's critical here for you to walk away with understanding and knowing is that we have two types of myelin that the body can go ahead and form. Both forms are made of cells. That's critical for you to understand. Both forms of myelin are made of cells, specific cells. And so when we talk about the myelin sheath, we're talking about individual cells that are wrapping themselves around the axon. They're wrapping themselves around the axon. How they wrap themselves around the axon determines how they function. And so structurally, Schwann cells and oligodendrocytes are different. And because structurally they are different, they behave differently functionally. What do I mean by that? Well, oligodendrocytes. Oligo here means few. So we're specifically talking about the oligodendrocytes. Oligo here means few. Dendro means branches. And cytes mean cell. And so oligodendrocytes literally means a cell that has a few branches. The way that this alters how it functions can be seen in the overall structure. Oligodendrocytes have a cell body. Right? They have a cell body. And they have a branch. Maybe that comes out this way. And maybe they have a branch that comes out that way. And maybe they have another branch coming out this way. And those branches are leading to axons of neurons. And what those branches end up doing is they end up wrapping themselves around one second. Change the color on that. So they end up wrapping themselves around the axon, creating the myelin sheath. Now, let me be perfectly clear. That blue structure is the cell body. That blue structure right there is the cell body. So notice with an oligodendrocyte, the cell body is separate from the myelin that's being placed on the axon. Compare that with what we see within what we see with Schwann cells. With a Schwann cell, this is what we say. The entire cell, nucleus and all, the entire cell is wrapping itself around the axon. One Schwann cell, one segment of myelin sheath. One oligodendrocyte, many sections of myelin sheath. Why is that so important to us? Well, it's important because, as we will find out within the next couple slides, 2 slides in fact, what this means for us is one of these cell types is more conducive, is more likely to lead to nerve regeneration. The other one is less likely to lead to nerve regeneration. And it all comes down to, basically, the lack of the cell body in the nucleus or the presence of the cell body in the nucleus. That leads to the ability for a nerve to regenerate itself. All right. So we already kind of talked about this already. Right? Schwann cell is going to, I kind of liken it to taking one of those animal balloons, you know, the things that the clowns use, and they blow it up, and it's really long, and then they wrap it up, and they make a dog and a cat and an elephant and a bird and a sword and all that kind of good stuff. If you take one of those balloons and start at the tip and wrap it around a pencil continuously on top of each loop is on top of the previous loop. So you just keep wrapping and wrapping and wrapping and wrapping and wrapping. And then when you get to the end, you take the end that's a little bit thicker, where you would blow onto it to blow up the balloon, and you tuck that underneath. And then you take another balloon and you wrap it 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 and you tuck the end. Leave a little space. Take another balloon, wrap it and 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 wrap it. That's how a Schwann cell or Schwann cells form the myelin. And so if you would actually cut, make a cross-section of a Schwann cell wrapped around an axon, it would kind of look like an onion. You would see the multiple layers of the Schwann cell that is present. With the oligodendrocytes, yes, the oligodendrocyte, the extension, is going to wrap itself multiple times around the axon. What you don't have, though, is the inclusion of the cell body. The cell body remains separate. Again, the myelin portions are formed by the Schwann cells or the oligodendrocytes, respectively. And then you've got the space in between each of those myelin cells, and that space is known as that node of Ranvier. All right? That's going to become critical for us on within the next couple of slides here. When we talk about nerve regeneration, understand that it is the peripheral nervous system that does lead to nerve regeneration. The central nervous system, under its own accord, currently does not have the ability to do nerve regeneration. It is only within the peripheral nervous system. And so my next question then becomes to you, what type of myelin was associated or is associated with the peripheral nervous system? And the answer to that is the Schwann cell. The Schwann cells are the cell type that is associated with the peripheral nervous system. It is the Schwann cells that do allow for nerve regeneration. Let me say that again. Schwann cells allow for nerve regeneration within the peripheral nervous system. And this little picture that you're looking at right down here kind of demonstrates how that happens. What we see going on, right here in picture 1, I'm going to label that picture 1, picture 2, picture 3. In picture 1, we have a normal function multipolar neuron that is innervating with the skeletal muscle, and we cut it. What you're seeing in this picture is the fact that the nerve has died. We've cut it. It's dead. In order for a nerve to regenerate, what happens within the peripheral nervous system is the last remaining Schwann cell that is intact and alive basically forms a tunnel. It grows, extends all the way down to where the axon used to be. So it goes from the healthy Schwann cell, and that Schwann cell extends all the way down, continuously, no gaps. That creates this tunnel that almost acts as a cast, if you would, or this protective tube by which the axon will grow down. Once the axon is completely regenerated, that elongated Schwann cell then breaks up into multiple new Schwann cells. Now, that is important to happen Because remember, we've got these little old things known as those nodes of Ranvier. And those nodes of Ranvier need to be there. We haven't talked about why they need to be there yet. But they need to be there. And so that one Schwann cell elongates all the way down to where the axon used to be, protects this protective barrier, all right, forms a tunnel. The axon regenerates itself down that tube of Schwann cell. Once it's regenerated, it can go ahead and that Schwann cell will then divide itself up into multiple cell types. The reason why Schwann cells work like that is because the nucleus is there. One nucleus, one section of myelin. With the central nervous system and the oligodendrocytes, you have one cell body that is controlling and regulating multiple branches of myelin connected to multiple axons. And that doesn't lead to successful nerve regeneration. That cell body's doing too much. It's got too many other things to worry about. It's not just controlling one little section. It's controlling multiple sections. And so that's why, you know, if you cut your finger, you're cutting nerves. You're cutting nerves when you cut your finger, especially if it's down into the muscle layer. If we did not have nerve regeneration, you would lose sensation every time you got cut. And we know that that doesn't happen. And so with oligodendrocytes, the key difference here, the key difference that I want you to walk away with is that they do not wrap their cell bodies around the sheath. They do not wrap their cell bodies around the sheath. And because of that, there is no regeneration. There's no neural regeneration. And this here is a pretty picture depicting the difference between the peripheral nervous system and the central nervous system and how the myelin is formed. Naturally, this one right here, the picture to the right, is the oligodendrocyte, whereas the picture to the left is the Schwann cell. All right. Let's talk about action potentials. First thing I'm going to say is that an action potential that is traveling down the axon is propagated or formed or created or maintained pretty much through the exact same process that sustains an action potential traveling around the sarcoplasmic reticulum leading to the T tubules. We don't see all that great of a difference. And so if you understand action potential generation within muscle, you will understand action potential within the axon. A couple of obvious differences. We've got this thing called myelin, which insulates the axon. We don't have that within the muscle. We have these things called nodes of Ranvier, which are action potential regeneration sites. We don't have nodes of Ranvier specifically within skeletal muscle. But we do have a very similar structure within muscle, and they are the voltage-gated sodium channels. And basically what the node of Ranvier is, very straightforward. The node of Ranvier is simply a voltage-gated sodium channel. Because what happens is the exact same thing that happens within the skeletal muscle. As the action potential is traveling down, in this case, the axon, as it's traveling down the axon, it comes to a sodium gate, a voltage-gated sodium channel. And that action potential forces depolarization to occur. Sodium rushes in. Sodium is a positive charge. The action potential is a positive charge. Positive and positive repels one another. Boom! The action potential now is very quickly going from the sodium channel down to the next sodium channel, passing through the myelin. And when it gets there, all right, before the action potential, arrives, that little old sodium gate is polarized. That is a polarized membrane. Right? Not only is it a polarized membrane, but we have achieved resting membrane potential. When the action potential arrives, depolarization occurs. Sodium rushes in. Your inside now of the axon is highly positively charged. We jump up to positive 30 to positive 50 millivolts. And so that's the other thing that I should mention here, is that it is polarized, and what we mean by that is we have reached or attained negative 70 millivolts. Right. We've reached negative 70 millivolts. And in order for us to go into depolarization, what that means is we need to, I'm going to come over on the other side here and type this out. We need to meet threshold, which we know is at negative 60 millivolts. So when the action potential arrives, depolarization has happened. What does that mean? We've met threshold, negative 60 millivolts, and voltage increases to a positive 30 to a positive 50 millivolts. That's when the action potential arrives. Where is it arriving? It's arriving to that node of Ranvier. There's the node of Ranvier, right there. The sodium rushes in when that action potential arrives. That depolarizes it. When the action potential moves on to the next piece of myelin, what happens within that node of Ranvier? Repolarization happens. Potassium leaves in excess. The voltage drops down to negative 80 millivolts, hyperpolarization, and then some of that excess potassium diffuses away, and you regain you regain polarization. You regain your negative 70 millivolts. And this just happens repeatedly all the way down the axon. So what we end up doing here is twofold, so to speak. What we end up doing here is twofold. The myelin serves the function of reducing the electrical charge. In other words, it helps to maintain the charge. It helps to maintain the action potential. Very, very thin surface area. Increases the speed, reduces leakage, reduces charge. But it also increases the friction. The node of Ranvier helps to decrease the friction, which helps to maintain the speed. 120 meters per second. 360 feet per second is what that sucker's moving. Right? This is a very, very fast moving process. But if we think about what is involved with that, every time you blink, every time you move, every time you breathe, anytime you do any of that stuff, the action potentials need to be able to move. And they need to be able to move when you need it to move and not when it gets there. And so it can't be slow acting. It can't be. It doesn't work that way. If you want a slow acting system, jump over to your hormones. Jump over to the endocrine system. That's slow movement. We don't want this to be slow movement. And so what does this all look like? Well, here we can see exactly what's going on. All right, and I'm going to type in using a nice little terminology here. All right, in the first picture, what we see here is resting membrane potential. Which means that the cell membrane is polarized. We are at negative 70 millivolts, and we're chilling. That's the first picture. The second picture what we see going on is action potential arrives. And when that action potential arrives, it leads to depolarization. Sodium, hold on one second here. Sodium channels open, and our voltage jumps to positive 30 millivolts. That's what's happening there in that second picture. You can see that the sodium is rushing in, and that automatically changes the electrical charge inside of that myelin. You've got the positively charged electrical current. You've got the positive sodium ions rushing in. Positive and positive doesn't mix. Opposites attract. And so when that positively charged sodium rushes in, changes the internal environment to positive, the positively charged action potential is there. Boom! That action potential wants to get out. Too much positivity going on. And so it speeds it up and sends it right on down that myelin sheath until it reaches the next node of Ranvier. Now, in the third picture, you see we have depolarization. Depolarization. Notice that the potassium is now starting to move outward. and this is actually behind just by a tad bit. Notice by the end of depolarization, the potassium is starting to rush inward. And when the potassium starts to rush inward, and draw another line, when the potassium starts to rush inward, we now enter into repolarization. And so this is our potassium now moving in. I'm sorry, that's the potassium now moving out. As the potassium moves out, notice we regain negativity inside of that axon. We repolarize it. We've reset that membrane, and it's now ready for the next action potential to come on. Now, what's happening during that repolarization? Well, we know that during repolarization, hyperpolarization is occurring, which means we've hit negative 80 millivolts. And we know that polarization will occur. And when that does, we return the membrane to resting membrane potential, which is what you are seeing in the last picture. Right? That's the return to resting membrane potential. And so that myelin, just to kind of summarize, it increases resistance because that myelin is insulating. It's not allowing that electrical current to go anywhere, so it increases the resistance. When you increase the resistance, you increase the friction, but in return for that, you're not losing any of the charge. You're not losing any of the charge. And so we have increased resistance, but we have reduced leakage. Right? We have reduced loss of strength of the action potential. Over time, if the nodes of Ranvier were not there, that buildup of resistance would definitely slow down the action potential. Without a doubt. It would slow down the action potential. And so what happens is those nodes of Ranvier are in there, and they're in there so frequently because what it does is it erases that resistance. It erases the resistance that's created from the myelin. And so it maintains that 120 meters per second. All right, real quick. We're going to blow through these last few slides here. There's not much more left. Trust me. So you can see here we've got different types of axons. Each of these different types of axons are going to do different things. The old favorite, typical, standard one is the multipolar, which again is right there. Right? It's got the cell body surrounded by the dendrites. The cell body is located at one end. The axon terminal buds are located at the opposite end. The axon is in the middle. Right? That is your typical multipolar neuron. Now, a unipolar neuron is what you are seeing right here. Right? With a unipolar neuron, what you are looking at is dendrites at one end, axon terminals at the other end, and the cell body is actually on the outside of the axon. It's not disrupting the axon, which is why we refer to this as a unipolar. It's got one fiber. At one end of that fiber is the dendrites. At the opposite end of that fiber are the axon terminals. And in the middle is the cell body. In the middle is the cell body. And that cell body is external. It's on the outside of the fiber. Unipolar neurons are said to be sensory. They are sensory. Whereas multipolar tend to be sensory and motor. What do I mean by that? Well, sensory neurons collect information from the environment. Motor neurons respond to that sensory information. And we'll talk about that arrangement here on the next slide. Sensory gathers information, motor responds. Multipolar have the ability to do both. Unipolar have the ability only to be sensory. They have only the ability to be sensory. Bipolar is called that because the axon, the fiber, is separated. The fiber is separated, and it is separated by the cell body. So you can see here, at one end you've got the dendrite, at the opposite end you've got the terminal end bulbs, and in the middle of that fiber you've got the cell body. That's why we refer to that as being multipolar. Or I'm sorry, not multipolar, bipolar, sorry. Now, again, bipolar tends to be sensory-based. Things like vision, things like smelling, things like hearing are all bipolar types of neurons. And then we have anoxic. Anaxonic, ana here means without an axon. This is basically a cell body that has dendrites with no axon. It is a cell body that has dendrites and no axons. And this here tends to be sensory-based as well. You'll talk about anaxonic neurons when you get into the endocrine system in INP2. Now, we talked about sensory and motor, and this slide here just basically breaks that out for you. What in the world am I talking about when I talk about sensory versus motor? Sensory neurons carry impulses towards the sensory nervous system. They're also referred to as afferent fibers, right? Whereas motor neurons carry that impulse away from the central nervous system, and they typically are referred to as efferent fibers. What allows sensory and motor neurons to communicate are known as the interneurons. What allows sensory and motor neurons to communicate are the interneurons. And so within the central system, you've got another neuron, and that neuron is receiving the information from the sensory neuron, and it's passing it off to the motor neuron. All right. Two good old terms that we've talked about in the past: parenchyma and stroma. Obviously, the parenchyma, the tissue that is doing the work of the nervous system, is the neuron. But the neuron cannot function by itself. As we know, think about this. We've already talked about two stromas. Right? We've already talked about two specific cell types that function in support of the neuron. The oligodendrocytes and the neurolemmacytes, the two cell types that form the myelin sheath. The myelin is there to support the function of the neuron. And so we've already talked about two of these, but when it comes right down to it, oligodendrocytes and neurolemmacytes are really examples of that fifth type of tissue that we've mentioned and never really discussed. Glial. It's glial tissue. Right? Remember, glial tissue functions within the nervous system, but behaves as either connective or epithelial tissue. And so that's what we need to look at here in the last 2 or 3 slides is these 5 types. Now, again, I'm not going to spend that much time on oligodendrocytes, neurolemmacytes, or Schwann cells because I think we've already beat that horse to death. But definitely we need to at least consider astrocytes, microglia, and ependymal cells. Astrocytes, astro here meaning star, cyte meaning cell again, so these are star-shaped cells. If you look, in the green, they are the astrocytes. The blue here would just basically be, this is a cross-section, so you're looking down, so that would be capillaries and that kind of stuff. It is only found within the central nervous system. It is only found within the central nervous system, and it basically serves as a link between the neuron and a blood vessel. And it creates, and this is something we're going to talk about more next week in lecture, but it creates something known as the BBB, the blood-brain barrier. Blood does not flow through the brain. It's external to the brain. What delivers nutrients and what removes waste within the central nervous system is something known as cerebral spinal fluid. Cerebral spinal fluid. So cerebral spinal fluid is kind of the blood of the central nervous system in a sense, because it's delivering nutrients and it's removing waste. What the astrocytes do is the astrocytes connect to the capillaries, and when they do that, the astrocytes increase selectivity, and they decrease permeability. And so they prevent things from leaking out of the capillaries. Right? They very tightly regulate what is entering into the brain and what is not entering into the brain. And that needs to be. The brain is very delicate. The brain is the center control of everything within the body. And if the brain gets damaged, you're up a creek. And so the brain has in place these protective measures that reduces the chance of injuring neural tissue. And that's what astrocytes really, in a sense, do. It forms a barrier between the outside world and the central nervous system, and the outside world being everything else within your body. That's not central nervous system, circulatory system, and endocrine system, everything and anything, anything and everything. Anything and everything. So, astrocytes are important. They function as glia. They are a stroma. They are an example of a epithelial type of cell that functions within the central nervous system. Oligodendrocytes, I'm not going to worry about that. We've already talked about it. Microglia, on the other hand, remember I made a big deal about this in lecture, is of the macrophage lineage. It's a cell type that cleans up debris. It breaks things down. It gets rid of dead tissue. It gets rid of invading bacteria, all that kind of good stuff. The reference I made to in lecture was, macrophages are their own lineage. They have their own steps involved in producing cells. Right. And the other macrophage that we talked about specifically was osteoclasts. Remember, we said osteoclasts are not of the osteoprogenitor cell lineage. They are, in fact, of the macrophage lineage. And so microglia and osteoclasts are both relatives. They're both from the same stem cell. They're both in the same line of formation. And again, the microglia here is going to form or function within the central nervous system. And lastly, we've got neurolemmacytes, which again we're not going to really function on, but we've got the ependymal cells, and ependymal cells line the ventricles of the brain. And so we looked at this in lab, the ventricles line, and I showed you basically where the ependymal cells would be. They line the ventricles of the brain. They line the spinal cord, so they're functioning again within the central nervous system. And their primary function is to produce cerebral spinal fluid, which again we'll get to in lecture. And so that's what I have for you for our introduction into the anatomy of the nervous system. Our next lecture will have us looking a little bit more in detail as far as the breakdown and organization of the nervous system, and really honing in on parts of the brain. And so, as always, if you have any questions, shoot me an email. And until then, have fun.